I am so excited to be here. I am a transplant to Utah, and I'm so fascinated by the landscapes in the West. And I get really curious when people say, I don't know. When there's this giant lake, 80 miles by 50 miles, and how many times have I heard, I don't know, about Great Salt Lake? So many unanswered questions, and unanswered questions are really what drives scientists. We seek answering questions. We really want to answer questions. So tonight I'm going to show you some of the answers that we had and talk about some of the work we've done, but also ask you some questions and ask for a little help. So I work... No, now it's not going to work for me. Oh, yes, it is. I just have to point it. I work in this body of water. Tell me, what's in there? Is it alive? Is it dead? What do you know about it? Yes? There's brine shrimp. That's right. And I have a picture here somewhere. I'm afraid I'm going to have to open the cabinet door. I think that's the problem. Bear with me just a second. Yes, it needs my signal to go around the cabinet. So there's brine shrimp in there. See, I was right on your wavelength, right? There's brine shrimp. They're mostly in the south arm. We do find them in the north arm, where I work in this lovely pink water up around Spiral Jetty. The part that's near Salt Lake City is really rich in brine shrimp. The cysts probably blow over the railroad causeway. We get some in the north, but not quite as many. Okay, so we have brine shrimp. What else do we have? What else do we know that lives in Great Salt Lake? Okay, we'll get there. Don't say that yet. Okay, yeah. Oh, and what are they eating? They're eating the shrimp. And what else are the birds eating? Brine flies. And brine flies don't swim, right? So when I say what's in the lake, it's not the brine fly, but it's this ugly creature. Have you ever seen those guys? Have you? They go like this in the water. Yeah, you're doing it. Right, everybody do this. Yeah, that's a brine fly larva. That's what they do. And they swim around the water. So you'll see those in the open water. Brine flies are kind of cool because, like a butterfly, they have this caterpillar-like form, this larva, and then they pupate on the bottom of the lake. So they're in the water, and then they go down to the bottom and form a pupa, like a butterfly would form a cocoon. And then that's going to float to the top, and then a fly emerges, and it's not even in the water. And then it lays eggs, and they're on top of the water. So the brine fly actually lives in parts of its life cycle in every part of the lake, which I think is ultra cool. But I'm going to get rid of them. Go away. Okay, so who said bacteria? Yes, thank you. It's usually everyone's last guess, so I like that you brought it up early. Bacteria, and there's another special word that might go along with bacteria. Thank you, yay! There's like one take-home word that I want people to take home tonight, it's that. The Archaea, I'm going to explain in a minute. Bacteria and Archaea are making these beautiful colors that you see in the north arm, these pink and red and purple and orange carotenoid pigment colors. The same pigments we have in carrots and sweet potatoes and watermelon. Whoops, geez, what happened? Okay, hang on. Technical difficulties. Okay, so how do we classify life? If I were to say, are you looking for something? If I were to say, give me a classification scheme, a mnemonic that you learned in school. Kings. Okay, King Philip came over for dinner. I learned kings play cards on Fridays, generally speaking. I was searching the internet to see what kids were learning today, and I found this one, which is a little different. I don't know why that's doing that. Okay. Which is Kings play chess on fine glass sets. Sounds very, very special. But I'm going to tell you from now on, you're going to say something before that. You're not allowed to ever say this again without putting a word like do in front, because I want you to realize taxonomy no longer starts with kingdom. And I'm going to say this in 2015. That changed in 1979. Okay? So if you were born since 1979, if you were ever in school since 1979, you should not use the mnemonic that starts with a K. Okay? In fact, we now have domains since 1979. A very famous scientist named Carl Woese discovered Archaea in 1979. He was sequencing genes. DNA sequencing technology was very new. And he started sequencing all these things that looked like bacteria. And he found two groups that were genetically different. And so if you look at their genes, completely different organisms. So he says there's 3 domains. And we know that to be true. The more organisms we sequence the DNA of, the more we know. Okay, so the 3-domain system looks like this. We've got eukaryotes, we've got bacteria, and we've got archaea. And the way the tree of life is drawn is by evolutionary relationships. So everything on that tree is alive today. It's not that we're going back in time when you see that tree, but we are building relationships of organisms. So if you were to see these, I know you can't read the little writing, but these are some algae, and we move into some plants and fungi, and, you know, lo and behold, humans, where are you? You are there. Okay, so you're there, and let's compare you to, I don't know, everybody's favorite laboratory pet, E. coli. E. coli's over here in bacteria. You're over here way far away from bacteria because you know you're genetically distinct from bacteria, right? You know that. But did you know bacteria is genetically distinct from archaea in the same way that bacteria are different from eukaryotes? So from this day forward, no one's allowed to just say prokaryote, because prokaryote is a meaningless word. It means without a nucleus, which refers to what you see under a microscope, which is 150-year-old science, okay? We have done some science in the last 150 years, and you're not allowed to use old words. Prokaryote was coined in 1923, and we've outgrown that word. And people say, well, what do I say if I can't say prokaryote? And what you say is bacteria and archaea. It's okay. We can separate them into genetically distinct places. So I'm going to tell you that sometimes grown-ups are afraid to use big words, but children never are. I want every kid in this room to say Archaea. Okay. Hey, there were some big kids. Okay, so this is the big word of the day. It's Greek, so it's got some funny vowels in it, right? This is the big word of the day that on Monday you're not allowed to forget. When you go to school, you're going to impress all of your friends. Okay? So you can see why people grouped them together. Archaea are the same size. They look similar in shape to bacteria. We know that they don't have a nucleus like bacteria. But you know, everything else is different. When you start to look at their genes, their biochemistry, their cell walls are different. And so we know that they're very different, even if they look the same under a microscope. Archaea comes from the Greek root. And this is it written in Greek. And does anybody speak Greek in here? It's Greek to me. So this is the Greek root, and it means ancient. When archaea were first discovered, people realized they were extreme organisms. They lived in crazy places like high heat in a hot pot in Yellowstone, okay? So if they're living in these extreme places, that's like primordial Earth. Yikes. I did that thing again. Okay. So primordial Earth, this is an artist's rendition of primordial Earth because nobody was there with a camera, right? So people sell Archaea live at these crazy places near volcanoes and thermal hot springs and high acid and high salt. They must be ancient organisms, and that's how they got their name. You might know that root from archaeology. It's the same root, okay, study of ancient history, right? So Archaea look like they're old, but the truth is this is the Earth they inhabit. They inhabit the same Earth that you inhabit. They are modern organisms, okay? They live now. Do they have a long history where they haven't changed? They've had as much evolution as we have, okay? Everything alive today has evolved. And we can't think of them as living on some crazy primordial Earth. No, they live on this Earth, okay? So the name is kind of a misnomer, if you will. However, it does tell you a little bit about who they are. So when I say the word extremophile, I'm often talking about Archaea. Although you might say brine shrimp are extremophiles. They live in extreme environments, and they're extremophile animals. Usually when people say extremophiles, they're talking about Archaea, but not always. So extremophiles, I mentioned, can live in really hot places. Those are called thermophiles. Thermo meaning heat, right? Extremophiles can live in really cold places. Those are called psychrophiles, and they're named after the Greek goddess Psyche, who was the goddess of wind. And so Antarctic cores, frozen glaciers, all of these have life in them. They have psychrophiles. Great Salt Lake. So these are extremophiles. However, they're living in an extreme of 30% salt, okay, at the north arm of the lake. Does anybody know how salty the ocean is in percent? Okay, in between, 3.4. Good. Good teamwork. So these pelicans, American white pelicans, are flying over Spiral Jetty and this group of researchers who are completely ignoring the cool pelicans. Both the researchers and the pelicans think, wow, what an extreme environment, right? But what are these archaea and bacteria living in here thinking? They're not thinking, wow, what an extreme environment. They're thinking, wow, I need salt to live. I hope this salt doesn't go away, right? So it's not extreme to them. It's not extreme at all to them. So they need salt to thrive in Great Salt Lake. Just like the thermophiles need heat and the acidophiles need acid, etc., etc. The ones that we have in Great Salt Lake are called halophiles. Okay, here is another Greek root, halo. The Latin derivation of that became sal, that became salt. Also celery and salad, lots of words came from salt. So halo is a root that means salt. They are literally salt lovers. Okay. So how might you find some evidence of Great Salt Lake halophiles? Some might suggest you use your nose. Some might suggest you do not use your nose. Your eyes. I'm going to show you how you can visually see halophiles without the use of a microscope. My students and I are often using microscopes and looking at them up close, or we're looking at their genes. And I'm going to say, let's stop calling them extremophiles, which is the big category of all the extremophiles, and let's start calling them halophiles now when we're talking about Great Salt Lake extremophiles. So if you use your eyes, you can look at the causeway that goes right through the middle of Great Salt Lake, the railroad. And tell me, let's see, my friend Victor. Victor, I need you. I just need you to look this way, Victor. Oh, no, he doesn't want to look. Is he out? Give him a minute. Hey, Victor. Victor, buddy. My buddy from last night. How you doing? Tell me, what color is this? I'm not sure. I'm not good with color. Oh, good. Good. Okay. What do you think that color is? That's kind of a weird color. What do you think it might be? Is it kind of green? Not really. No. Is it kind of brown? No. What do you think? Who thinks that the color on this side, and colorblind people, I'm a geneticist, so I know who you are and what proportion in here should be colorblind. Colorblind people don't answer this question, but everybody else, do you see a different color on the sides of those causeway? Yeah? Okay. You just identified two different microbial communities. Okay? Microbial community on the south part of the causeway has a lot of different algae species. There's a lot of photosynthesis going on. A lot of bacteria, some archaea, but not as much as over here. The Halophiles, the Halophiles, are loaded with those red and orange and purple pigments. So they're making this beautiful color, whereas this is a lot of photosynthesis going on over here, a lot of chlorophyll, okay? So different colors, yes? We were talking about this today with the grad students. Someone asked the question, the causeway was built between 58 and 61. Before that, it was a trestle bridge where the water went underneath. When they started rock filling the causeway and preventing exchange, this started happening. But if you look at the historic salinity data, there are times, particularly when the lake was low, like in the mid-60s, there are times when the whole lake was at about 23% salt. I'm shocked that it did not turn pink. I think there were probably parts that turned pink that no one paid attention to. I have gone back in historic journals. I've looked through fur trader journals and pioneer journals and looked for any reference of the lake being pink, because that's what happens all over the world in hypersaline bodies of water. These halophiles have these carotenoids, and the water turns pink. Nowhere can I find a reference of Great Salt Lake being pink. So I think it never got to 25, 28%, or we would have seen that bloom of halophiles. I talked to my friend who works on the Dead Sea, and I asked him, like, do you have historical references? Because the Dead Sea blooms pink, and then that bloom goes away, and then it blooms pink again. So there are times in history when it's pink and times when it's not. And so I asked him about this, and he said yes, and he quoted the Bible verse. So if you're a scientist working in Israel, you get to use the Bible as a scientific text, which I thought was great. So there actually is a reference to the Dead Sea having a red coloration, and it's unknown as to whether that was a sunset metaphorical depiction or whether... So anyway, I have actually looked for some kind of historical evidence. So, no, this is artificial, and this dichotomy did not exist. This pink water likely started in the 60s, and that's what we know. So it's kind of losing its... Okay. So you were using your eyes to see microbial communities. Now, imagine, this is a purple sulfur bacteria. It's giving off sulfhydryl gas. Close your eyes and just imagine what that smells like. Yeah, it's pretty stinky. Sometimes you can turn over some sediment in Great Salt Lake and you release what we know of as that lake stink, right? So the next time you smell Great Salt Lake, I want you to think about it differently. Think about you're sensing microbial communities, okay? It'll make you happier and it'll make you feel more scientific. Don't say, ooh, yuck. Say, ooh, life, I smell life. It'll make you feel better, okay? I I also wanted to point out in this picture that my colleague Jamie took around Hat Island, you can see cyanobacteria growing on these calcium carbonate stromatolite structures in the lake, and those guys are doing a lot of photosynthesis and bringing some energy to the lake. So again, pigments, you can really see presence of organisms. What about these salt crystals? Do you think anything is growing in these salt crystals? Yeah. If you were to just pick up that salt off the shelf, you could probably sell it for a lot of money and sprinkle it on, your salad in a fancy restaurant, right? You could call it pink and green sea salt from France, and it would sell. But really, those are halophiles, and these are some halophilic algae that are living and producing energy right on a salt crystal. Okay, these microbes can be dried up and rehydrated later. We actually have been looking for them in ancient salt deposits, which has been very interesting, and I'll talk about that in a little while. So if you see water like this, you better believe something's living in it, okay? And it's not brine shrimp. This is too salty for brine shrimp. These are our halophilic Archaea. They're making beautiful pigments. This is a salt pond at Cargill on the south arm of the lake that just had a beautiful year one year. Okay, so if we were going to take salt crystals from a nice little pink puddle and try to grow organisms out of it, I wanted to simulate such an experiment. So if there are kids here who want to come up and simulate an experiment, help me do a demo. Would you like to do that? Anybody here? No, you're shaking your head no. Why are you shaking your head no? Come on, help me. Would you help me? All right. Do you have money? Right there. We have brine shrimp tattoos for anybody who wants to participate. Yes! Look at her go. Come on down. Come on. All right. Victor, come down here. What are you doing? High five. Yes. All right. High five to you. Nice. Do you want to come, Victor? Okay, you guys get to wear gloves. Yeah. Does anybody else want to come now that you get to wear gloves? Do you want some gloves? Some? Okay. All right, you can take the gloves home. You're not going to get them dirty. Okay. You can make glove chickens after this is all over. Do you know about glove chickens? We'll teach you about glove chickens. Yeah.
Are we young enough to do this?
Yeah, sure.
Okay, good.
You might have to help herd, though.
Okay, we'll herd them in.
Have you ever herded cats? Okay. All right, pass these around, and I want you to... Do you need help? Victor, I'm going to help you. You got it? Here, you hold the bag. All right, Victor, let me help you.
Okay.
You ready? Okay, I'll hold it up. Okay, you get your hands in. Wow! It's a little big on you, huh?
Yeah.
That's okay. Do you mind? Okay, ready? 1, 2, 3. Awesome. Okay, now go like this. Sorry. That's what you get. Okay. Now, where are my nerd herders? Nerd herders? Yeah. You're the nerd herders.
Okay.
Okay, you guys have to make sure everybody picks out a salt crystal. Do you want to pick out a salt crystal? Okay, you take one and then pass it around. Everybody can take a salt crystal. Oh, that's good. All right. Okay, so now this part, that's okay, Victor. Here, it's hard to do with those hands. You got chicken hands. You got chickens on your hands. Okay, ready? You want to pick one out? Awesome. All right, did everybody get one? You like that? All right, did you get a crystal? Ooh, pretty. Yeah, those are very small. I didn't anticipate the children being so large. Okay, so we're going to replicate an experiment. And it's kind of like a cooking show. I've actually already cooked these. So we're just going to replicate an experiment where what we would do in the lab is actually take these crystals and roll them all over a plate. But I'm not going to have you do that. Will you just show how you would roll it? Like pretend it's open. Yeah, we would do this, right? Yeah, you want to do that? Yeah, there you go. Okay, you do it too. Very good. Okay, how about you? That's what we do in the lab. We would open this up and we would rub our salt crystals on it. But I don't want to take your salt crystals away from you because now you can take them home. Okay, so you guys take them home. No, you can. Yeah, you can totally taste it. You don't mind brine fly poop, right? Oh, environmental samples. It's nothing dangerous. Okay, thank you. Thank you, guys. Now I'm going to pass around the results. Go ahead. You can have them. Okay, there's good. You can have it. Take it home. Oh, yeah, brine shrimp tattoos, everybody. You folks come back. You got to have those. Yeah, you might need the tramp stamp version. I'm just kidding. Just because you're older, you get the bigger ones. We call them that for our college students. Okay, awesome. Okay, so I'm going to pass these plates around so that you can get an idea of what these halophiles look like. One of the reasons I use halophiles is I work with undergraduates and sometimes I work with high school kids, and I like to use a model that are pretty much protected from my students, right? You thought I was going to say it was the other way around, right? These are totally safe. These plates have 23% salt on them. So nothing from you that grows at 0.1% salt, 0.01% salt in your cells, nothing from your body can grow on these plates, right? So if you open them and you breathe on them, it's not going to hurt these. If you were to eat these, which I don't suggest you do because they wouldn't taste good, it wouldn't hurt you because these can't grow inside you because you don't have enough salt for them, okay? So they're very, very, very safe to use with students. So we love to work with teachers and give them access to halophiles because they're very safe to use in the classroom. I used to teach microbiology, and I just found it really scary to be working with things that could be pathogenic. Okay, do you want to hand these out kind of distribution-wise? So you can take the top off so you can get a good look at the colonies, and don't be afraid of them. They can't hurt you, okay? Alrighty then. Thank you, everybody, for helping with that experiment. I really wanted to show the kinds of things that we might do to grow some halophiles in the lab. We do see some beautiful evidence of bacteria in Archaea. Almost exclusively out of salt crystals, we pull out the Archaea. And we find all kinds of different organisms, colonies of different sorts. For several years, I've been doing this in my genetics class, and all of my students have been isolating their own organisms out of salt or out of a different pot of water from different parts of the lake. And they each get their own organism, and they learn a bunch of genetic techniques, and they genetically identify it by sending their sequencing to USU. And I'd like to have a shout out to Ninglin in the audience here who does all of our DNA sequencing, and I'm so grateful. I'm so grateful. So it's been a wonderful collaboration, and so many students have learned so much. I have about 50 students a semester in the fall who are learning genetics, and they're all sophomores, and you help them get excited about genetics, so thank you. Okay, so we also pull out some algae from the lake. This is Denaliella salina. working with a collaborator, Jürgen Pole, who's at CUNY Brooklyn. He has found at least 7 subspecies of Denaliella viridis, it's called, in the south arm of the lake. So if you think about it, we see all 3 domains of life in Great Salt Lake. Even in the north arm, where life is a little more restricted, we see some algae there. We see we now have 12 species of fungi from the north arm of Great Salt Lake. So we see a lot of things in the eukaryotic realm that we didn't anticipate. We see Archaea, a whole bunch of it, hundreds of different species, and we see some bacteria that are there. So all 3 domains of life are represented in this crazy extreme environment, which is a fun story. If we want to look with our microscope, we can look a little more closely. This is mimicking a light microscope. We might make a slide and look at it in the lab really quickly. We did some experiments with electron microscopy, where we can really zoom in, not using light to go through the specimen, but using electrons, and you can get really close. So you can start to see the shapes of the cells of these archaea are similar to what we're taught are common shapes with bacteria, spheres and rods. But we also see some snakes. Where's my herpetology friend? Does that look like a snake? No, that's a microbe. We also see like these revolving pyramids. That one's kind of flattened, but some really interesting, we see squares. This is a cool story. In every single high salt place on the planet, we have square microorganisms. Nowhere else on the planet, okay? They divide and they don't let go of each other. They divide in big sheets. And they're all genetically related. So figure out how that genetically related organism is the same in a salt pond in Germany as it is in a salt lake in Australia. Crazy story. These cells that we were zooming in on, we started to notice these little lollipops. Does everybody see the little lollipops? I love that virologists actually call these lollipops. These are head-tail viruses, so that just refers to their shape. Those viruses, do they look scary? Anybody know what a virus is? Anybody ever get the flu? You ever get the cold? Ever have a virus? Okay, well, these viruses in the north arm of Great Salt Lake, they won't hurt you because they're only after one thing. They are the predators of these archaea, okay? It is a predator-prey relationship, and the little guy is in charge, all right? So we actually have done a lot of characterization just from microscopy, but not genetically, of these viruses that are in Great Salt Lake. And they're absolutely beautiful and diverse in structure. We have some that are shaped like lemons, which actually are also found in salt lakes in Australia, which is really crazy. We have viruses that are these sort of traditional head-tail. Some of the first viruses that were ever discovered were in that shape. We have spheres. We have a lot of different shapes of viruses, but if you think back to that other picture, they're much smaller than these already very tiny archaea, right? So I've just blown it up here so you can see it big. So the viruses are there. They're helping modulate the archaea who are present. So the next time you're out at Spiral Jetty and you're wading around in that salt crust and that water, or you're swimming, come back. Or you're swimming in the water, think about the microbes that are swimming all around you, okay? It will make you happy. So now you're going to be one with the microbes. When you smell the lake, you're going to think about the microbes, and when you step in the water, you're going to think about the microbes. This is what our offices look like at Great Salt Lake Institute. This is an ultra cool experiment that we did with a 4th grade classroom. It was my daughter's class, and now she's in 7th grade, so it was 3 years ago. And we we took these spaghetti containers that you can buy at Shopko for $3, and we gave each kid, or each group of kids, a spaghetti container. And they basically, if you try to order what's called a Winogradsky column, it will cost a couple $100, but you can actually do this in a spaghetti container. What's important is that you put the sediment in, the sand, as well as the water. We did this in different spots around the lake with different groups of kids. And you can see some of the North Arm Halophiles, for example. Actually, this one is from a really strange area. Does anybody know about the Bangalore pumps? Some people do. Jamie took that sample out there. Is that the one, or is it? Oh, it was this. It's that biofilm one, isn't it? Yeah, that's the biofilm one. So all of these are from different areas, and the trick is to put sediment as well as water and watch the microbes go crazy. And you can see like this pink, this is that purple sulfur bacteria I was telling you about. It makes a perfect line under the sediment. This is the water column with the filamentous algae. And you can watch over time, over the years that we've had them, they completely change. So for teachers to have these in the classroom and watch the microbial communities change over time, it's absolutely beautiful. And I think we're doing a little bit of art. What do you think? Yes, Mark? No. We thought we could, actually. And I think you could do it so that maybe it's more predictable. We thought we could, but it turned out that one of these pink samples, and I can't remember which one it is actually from the South Arm, where none of the water was pink. And so that surprised us. The formation of this biofilm, you see how it's kind of peeling off like a sheet from the inside? That biofilm was bright purple, and then Jamie moved it where she had it in her office to another spot, and it turned emerald green. And then it peeled off. And so it's clearly a community made of different organisms. It's a cool biofilm, and they were all talking to each other, and they're like, hey, we like the sunlight here. And then the other organisms probably did fine in the different location, and then they just ran out of oxygen or whatever. So you can really have a good time. Just with spaghetti containers and a shovel. We've also done some work with salt. This is actually a 253 million year old piece of salt that I got from New Mexico, not from Great Salt Lake, from an underground salt mine where I sample sometimes. And we did some experiments where we looked for not cells, but actually biological molecules. We found DNA, we found cellulose preserved from the age before the dinosaurs. So that's a really cool story. But a lot of the salt experiment is much like what we were looking at, the simplified version that the kids did up here. And we grow all kinds of amazing organisms out of there. So I want to start finishing off here so we have time for questions. Just by asking, does life have limits? Is there a too hot? And the answer is yes. It can be too hot. 100 degrees in Celsius is boiling, and there is a life form that is called strain 121 because it can go to 121 degrees Celsius. It actually can live, I think, for 2 hours at 130 degrees Celsius before it dies. But 121 it can live. It can live and divide at. So if you look at heat, if you look at cold, if you look at amount of radiation exposure, there is an upper limit and a lower limit to each of those things. With salt, you can't get more salt in the water to please these organisms. Okay, there's no upper limit except that the water can't hold any more salt. So with salt, it's not really that limited. So we used to say things like, oh, life could never exist on Mars because it's too cold. Well, now we know about life that can exist at those temperatures right here on our planet. We used to say life can't be on Mars because of the level of radiation. We now know about organisms that can handle huge amounts of radiation, okay, on our planet. We used to say life couldn't be on Mars because there wasn't enough water. Now we know about organisms that can live in desiccation and cycle in cycles of desiccation. So extremophiles have taught us to open our minds and to think beyond our planet. Whoops. So Mars is a place that I'm very interested in. I'm funded by NASA right now to do work with getting molecules out of salt, thinking about could we design something for a rover. The reason I'm really super interested in Mars is that there's a giant Bonneville salt flats on Mars. There's an area called Meridiani Planum that the first twin rovers, Opportunity and Spirit, Opportunity found that there's this great salt deposit. And on Earth, we know that that's associated with water. If we see a big salt deposit, we know it used to be a sea. We call it, actually, geologists call it an evaporate, right? Meaning that water evaporated. So we figure that this spot on Mars was actually a lake that became a salt lake that now is a salt deposit, right? So if we can find life preserved on Earth in ancient salt deposits, maybe that could be where life could be preserved on Mars. So millions of years ago, when there was an atmosphere on Mars, and now geologic evidence tells us clearly that there was water on Mars, big water, big seas. Why wouldn't we think life could also exist there? Now, did it exist there? We don't know. But if it did, I would want to look in the salt and see what's preserved in the salt there. So that's one of the reasons I like to look to the sky when I think about extremophiles. So there's the current rover there. Who knows the name of the new rover who's on Mars right now? Curiosity. Thank you. I love that name. Curiosity is the best geologist ever invented, and I don't mean any disrespect to any geologists in the room. Curiosity is sending back more data than any graduate student ever produced. And now the question is what to do with all the data. The data is public access. It's really amazing. I think we're getting, that's why you're seeing reports or Facebook links or whatever. Every week, perchlorate was discovered on Mars last week. Every week there's a new story. So please stay tuned to Curiosity. We are learning so much about the potential for life off our planet. So I'll leave you with a picture. This is my son and Jamie's son playing out at Spiral Jetty when the water was a little bit high. And I just want to remind you that those are extremophiles, our children. They've grown up in the lake. They're very extreme. But you're swimming in a pool of microbial soup that can't hurt you, just like the plates I sent around, okay? So enjoy the lake, engage with the lake, experience these extremophiles of Great Salt Lake. And that's all I have for you. Great, thank you. I'd be very happy to answer any more questions that people have. Yes, Ned.
Earlier today in your presentation, you showed a picture of the lake and you showed the dolphins and the salmon. But at the very bottom of the south end, it looked like it was pink green. Is that because the water level is so low and we're getting high salinity at the very south end?
No, what you were seeing were salt ponds. In fact, I didn't take too much time. I had a slide at the end that I didn't show because I didn't want to go too far. But the salt ponds basically dam up some of the water and use evaporation and create hypersaline portions. And so I should have said that sometimes when you fly over Great Salt Lake and you see pink areas, sometimes those are dammed up salt ponds. And I had my one slide, which I'll have to share with you, Brandon, that I didn't show today earlier, was the salt ponds next to an early Piet Mondrian. They're very similar. They're very similar. In fact, has inspired some artwork from some of my students. Yeah, if you see a pink color, you know there's salt saturation going on. And that is the Magcore pond that they share with Morton by Stansbury Island. I think that's probably what you saw. Yes? Extremophiles. Archaea. Yeah. Thank you. Big word of the day. You got to impress people on Monday. Yeah. You're bringing up a couple of really good points that you maybe even don't know. Yes. What do they live on? Number one, we don't know this giant picture about Great Salt Lake carbon cycling. We don't understand it. Most ecosystems had that figured out 50 years ago. We don't know this basics of carbon cycling in the lake, which is kind of sad. And the lake is so multifaceted that it's a really hard experiment to do. So we don't know how the nutrients turn over. But we would suggest that microbes are doing that job, and they're breaking things down and turning over the nutrients, because we know that's what microbes do in every ecosystem. So there are nutrients free-floating in the water, and they're certainly infused with that. Some of them, however, can use sunlight to produce some free ATP. They have salt pumps that they can use the energy from sun. So they're not truly photosynthetic, the Archaea. And then some of the cyanobacteria and other eukaryotic algae are bringing in sunlight energy. So there's definitely a lot of inputs to the lake. And Giovanni, who works here, and I were talking about this yesterday, about potential carbon input and how we really don't know what all the inputs are to the lake. What about the dead brine shrimp? You know, they die in the fall, right? That's a lot of chitin that goes into the lake. But microbes would have to digest that and turn it over into nutrients. So we think that microbes are just sort of ingesting the nutrients in the water, but they're also involved in sort of breaking the whole business down and turning over the nutrient system in the lake. But no real data on that. That's the sad part. Yeah. Other questions? Yes, sir. Yes? Oh, interesting. Right. Right, that you take it. Yeah, you know, the biggest confounding principle in medicine is how to deliver the technology or the drug in a way. I mean, because you think about your stomach is going to, speaking of extremophiles, your stomach is pH 2. That's like vinegar, right? Those are acidophiles in your stomach, by the way. Yeah, but it's got to make it through your stomach, right? First of all, and so there's some really cool tricks to drug delivery, but it's not simple. So I would say you'd probably want to break it down to what proteins are functioning and try to find a way to deliver those. But that's a really cool idea, and that's the way a lot of good ideas start. Yes, sir. Yes. Yes. Yeah, it really is. In fact, we're out there a lot, and we see mostly Europeans swimming in the lake for some reason. Utahns aren't there. So it really, if you think about it, the things that you should be concerned about are the things that grow at 98.6 Fahrenheit and live in your nutrient conditions, right? Those are the things that you should worry about inhabiting you. E. coli, salmonella. The reason salmonella is so scary is it's an intestinal bacteria. It knows your system, and it knows how to live there. So if you have a bad strain of salmonella, which of course is, running around the chicken industry, then it already knows how to inhabit you. Halophiles can't live in you. If you put them in water, what happens is the water rushes across their membrane into their cell to try to dilute the salt, and they pop. So you can't, eat them. Yeah. Oh. It's actually an algae. It's actually, some snow people here probably know the answer to what species it is, you know? Oh yeah, it's a Chlamydomonas. That's right. I remember that now. Yeah, okay. And it's so pink snow is known as this red algae. Yeah, carotenoids though. That's what makes it pink. Yes.
What about worlds beyond Mars?
Yes.
There's really cool stuff with some of the European and etc.
Yes, absolutely.
What's your take on that?
So I've been working, I've been talking to a lot of people since I've been plugged into NASA, which has been really fun. And I've met with people who do imaging, and I've met with people who do remote sensing through lenses that can filter and detect things like CO2. And there's some amazing technology. So one of the things that happens on our planet, it's called the carbon cycle. And biologists like to own the carbon cycle, and then you talk to earth scientists and they like to own the carbon cycle. And it's something that goes between the biosphere and the lithosphere, right? So it has to have tectonic plates to work. And I realized this when I read an essay a few years ago. If you don't have tectonic plates on a space body, you can't have life. So there are places that become really interesting, like Titan, one of Saturn's moons, because it has ice sheets that have tectonic activity. Mars doesn't move anymore. The plates are frozen, but they used to be there. So I teach a class called astrobiology, and one of my students came up with a grant proposal to implode missiles towards Mars and try to get its tectonic activity moving again, which I thought was hilarious. So that's one thing I would look for, and that's one thing that scientists look for. They can remote sense gases that are associated with life. You can look for presence. The geology with these rovers on Mars, by the way, is almost at the resolution of geology on Earth. Okay, we can almost do the same geology on Mars that we can do on Earth, which I think is astounding. So if we're remote sensing from a satellite, it's not quite that good, right? But it's still good. And so we're learning, that's how we're learning so much about the Jovian moons, the new data that's coming back from there. It's clear there is fluid, there's water. It's clear there's water elsewhere, even in our solar system. And there are reasons to look at moons as opposed to planets, because moons do this tidal heating thing as they orbit. They actually have a frictional component where they expand and shrink. And you thought I was just a biologist, right? I ended up on a physics thesis committee for some reason because I was an astrobiologist. I learned so much about tidal heating. But there's reasons to look at moons versus planets too. So some really cool stories out there, and we're living in an age of excitement, and I think we will very soon have the answer about, are we the sample size of one life evolution in the universe, or is there more than one evolution of life in the universe? So I'm excited. It's a good time to be alive in science.